So today the first lecture will be introduction to the field of immunoengineering. I have been invited by Young Pharmaceutical Scientists Association to give this talk, and there will be a series of talks about immunoengineering. Today is the first one, the introduction part. So what is the definition of immunoengineering? Here, when we talk about immunoengineering, we talk about using innovative engineering tools to modulate the immune system for design of immunomodulatory therapeutic strategies. So if we have a problem or certain disease related to the immune system, and we need to modulate the immune system, modulate mean we need to stimulate or we need to suppress. Stimulate like in case of cancer and suppress the immune system activity like in case of autoimmune diseases. So let's generally talk about modulating the immune system. And when we are trying to modulate the immune system using certain engineering tools, this is what we call immunoengineering. So what are those engineering tools that we can use to modulate the immune system? We can use cell engineering. So we will talk about cell therapy like CAR T cell, for example, and adoptive cell therapy. We will use also tissue engineering that we can construct certain organ maybe in vitro or by 3D modeling or by 3D by printing or organ on chip microfluidic model to replace or to correct the function of certain organ or certain tissue in the immune system. Of course, also we use the tools of drug delivery, nanoparticles and other than nanoparticle, even microencapsulation, anything related to drug delivery. Engineering nanomedicine, we can also synthesize certain biomaterials that can modulate the activity of immune system. So when we use the term immunoengineering, we mean that we use some tools like drug delivery, like cell or tissue engineering or biomaterial synthesis, polymer synthesis, 3D by printing, microfluidic chips. All these engineering tools, we exploit these tools to modulate the activity of immune system. And this will lead to design of certain immunomodulatory therapeutic strategies. When we focus on cancer immunoengineering, and this is my focus, so here we will use those engineering tools and immunomodulatory biomaterials for modulating the immunosuppressive tumor microenvironment as a way to help the immune system to kill the tumor cells. So our problem in case of cancer is that the tumor microenvironment is immunosuppressive in nature. And we will talk about the reasons for this immunosuppressive tumor microenvironment. And our job here is to stimulate the tumor microenvironment or to reverse the phenotype of tumor microenvironment from immunosuppressive phenotype to immunostimulant phenotype. And this is will be achieved using certain engineering tools. We will use, for example, here some cells that we can engineer the surface of cells with some nanoparticles. These nanoparticles can be loaded with DNA. For example, in case of CAR T cell, we transfect T cells with certain DNA encoding certain tumor antigen, chimeric antigen receptor. Or some nanoparticles loaded with cytokines to activate these T cells. Or also we can make combination between different modalities like radiation or chemotherapy or phototherapy together with immunotherapy to treat cancer. All these trials is to stimulate the immune system to fight cancer using certain engineering tools. These engineering tools can be nanomedicine, can be some antibodies also. This nanomedicine also can be not only attached to the cells or used to engineer the cell surface, but can be loaded with some stimulatory molecules, cytokines, toll-like receptor agonists to activate dendritic cells. Also, it can be loaded with TGF-beta inhibitor, any drugs that can modulate the activity of immune system. Our target here is to reverse the tumor microenvironment from immunosuppressive state to immunostimulant or immunoresponsive state. Or you will find in some textbooks that we need to reverse the tumor from immune cold to immune hot. It's the same meaning. immune cold, that mean immunosuppressive tumor microenvironment, to immune hot, that mean immunoresponsive or immunostimulant tumor microenvironment. In all cases, our target is to make the immune system in active state to fight the cancer cells. And when we use engineering tools to do this, this is what we call immunoengineering, or in this case, cancer immunoengineering. In some cases also, we can coat the nanoparticles with some cell membrane, like the membrane of T cells, or for example, also to fight, or what we call the biomimetic, to fight the cancer cells. All this we will study in details in our talks about cancer immunotherapy. But we need to focus first on the basics of cancer immunotherapy. So all of us know what is cancer. Certain population of cells start to grow and proliferate without stopping. This is the main characteristic of cancer, right? And by time, these highly proliferating cells will start to spread into surrounding tissues and forming metastasis. Okay, so if we talk about the history of cancer therapy, at the beginning, all the people know about cancer is that some abnormal cells with uncontrollable proliferation start to appear in and invade certain tissues. So the main characteristic here is uncontrollable proliferation. And here, the first drugs discovered to treat the cancer is chemotherapy or cytotoxic agents that we use to kill these highly proliferating cells. And the main idea that we use some drugs that affect those cells with uncontrollable proliferation or very high rate of proliferation, and those cells will be for sure more affected by these drugs rather than the normal cells with normal rate of proliferation. After that, people start to realize that this uncontrollable proliferation come from or as a result of accumulation of genetic and epigenetic mutations in the cancer cells. And here the concept of mutation come to our mind. And in response to this mutation, we designed what we call the targeted therapy, or the era of personalized medicine. And here we will start to focus about the cells, only cells that express those mutations, and we use drugs to correct these mutations. And of course, those cells that have these mutations will be more affected with the target therapy. That's why we call it targeted therapy. So targeted therapy target only cells that express some genetic or epigenetic mutations leading to formation of cancer. And in phase 3, researchers started to realize that the cancer is not only a genetic or epigenetic mutation characterized disease of cells, but also, or in fact, it's a constant struggle or battle between cancer or malignant cells from one side and immune cells from the other side. And here come the importance of immunotherapy. So the immunotherapy here is dependent on some immunomodulatory strategies to make the patient's own immune system able to fight against the cancer. So in contrast to chemotherapy and the targeted therapy, we use exogenous agent to attack the cancer cells directly. Here in immunotherapy, this is not the case. Here in cancer immunotherapy, we use some immunomodulatory strategies like antibodies or any drugs that activate first the immune system, like, for example, cytotoxic T lymphocytes. And the patient immune system or cytotoxic T lymphocytes, for example, the main effector cells, will start to attack the cancer cells and kill the cancer cells. This is the concept of cancer immunotherapy. So here, we only potentiate or stimulate or activate the patient immune system in order to be able to attack and fight cancer cells. Like we see here in this schematic is the immune cells, T cells, this is in red color, attack the cancer cells in white color. Okay. And we know that in recent years, there is much more focus and huge growth in cancer immunotherapies. And for example, the number of active clinical trials on cancer immunotherapies has increased by 33% from only 1 year ago. And this is very high percentage. So yes, this is the era of immunotherapy, and all the focus will be directed to immunotherapy rather than chemo and targeted therapy by time. Here we just need to focus on the 3E hypothesis of cancer immune system interaction. In the first phase, or the initial phase, elimination of malignant cells by innate or acquired or adaptive immune effectors. So here the immune system, by its two arms, the innate and the adaptive, we will talk about this in details. First, the immune system is able to attack the cancer cell and kill the cancer cell. This is what we call elimination. So elimination of malignant cells, that means that our immune system in the beginning has full activity and is able to recognize and attack the cancer cells, leading to their elimination or killing. By time, cancer cells will try to establish equilibrium between the machinery of immune system. So cancer cells here try to evade the immune system, and the immune cells still try to capture the cancer cells and attack. So here there is equilibrium, no winner in this phase. But unfortunately, in the final phase or the third phase, which is escape of cancer cells from immune control. And this is the fatal phase. Here, the immune system unfortunately become unable to attack the cancer cells. There is many reasons for this resistance, what we call immune resistance, that enable the cancer cells to escape out of the control of immune system. But mainly, cancer cells do two things to evade or escape the immune system control. Number one, selection of non-immunogenic cancer cells, or what we called immunoselection or immunoediting. Here, the cancer cells try to be non-immunogenic. Non-immunogenic, what is the meaning of non-immunogenic? This means that immune cells will not be able to recognize the cancer cells. The first step of the immune response, that immune cells try to recognize the cancer cells, and this requires the cancer cell to be immunogenic. Immunogenic comes from expression of certain tumor-associated antigens or new antigens on the surface of cancer cells. These antigens can be recognized by antigen-presenting cells, like what we illustrate later, like, for example, dendritic cells. And here starts the immune response. But if cancer cells succeeded to make themselves non-immunogenic, for example, by down-regulation of these tumor-associated antigens, here the antigen-presenting cells and the immune system will not be able to recognize the cancer cells. And the net result that cancer cells will evade or escape of the control of immune system. Not only this, cancer cells also tend to generate immunosuppressive tumor microenvironment. So cancer cells not only downregulate its antigenicity and the expression of tumor antigens, but also secrete some factors. These factors can modulate all types of immune cells present in the tumor microenvironment and convert them to immunosuppressive phenotype. That means even if the T cells present and infiltrate the tumor, it will not be able to attack the cancer cells because its activity is suppressed or exhausted. So cancer cells in the final phase try to escape the immune system mainly by two pathways or two mechanisms. The first one is to be non-immunogenic, non-antigenic, so that the immune system will not be able to recognize these cancer cells. And number two, cancer cells tend to induce suppression of immune tumor microenvironment. Need to suppress the activity of all cells present in the tumor microenvironment, including dendritic cells, T cells, macrophages, fibroblasts. All the tumor microenvironment will be in immunosuppressive state. And here comes the importance of cancer immunotherapy. So cancer immunotherapy, what is the target of cancer immunotherapy? Cancer immunotherapy will try to reset the relationship between the cancer and the immune system from escape to elimination. We will try to reverse this third phase of escape into elimination. We will try to stimulate the immune system again and reactivate the immune system by different strategies and using different engineering tools to make the immune system again able to eliminate the cancer cells. And here we will just try to remember some basics of immunotherapy. So of course, the immune system has two arms: innate immunity. Innate immunity, this is non-specific and the first line of defense against any pathogen or any cancer cells. And this can include dendritic cells, macrophages, natural killer cells, neutrophils. And this innate immunity, although non-specific, but will lead ultimately to activation of the other arm of the second arm of immune system, which is adaptive response or adaptive immunity or acquired immunity. This is the specific type of immune system. The specific arm of immune system includes mainly B cells and the T cells. And we know that T cells, the effector T cells, were by its two types, CD4 and CD8 positive T cells, will be able to attack the cancer cells, especially CD8 positive T cells, while B cells will be able to produce antibodies against also these cancer cells. So if we talk about the T cells, we have four main types: CD4 positive, the helper T cells; cytotoxic T cells, CD8 positive, or what we call cytotoxic T lymphocytes; T regulatory cells or suppressor T cells, or sometimes it's actually one type of CD4 positive T cells, but has certain characteristics and has expression of unique transcription factor FOXP3, and memory T cells, and this can be CD4 and CD8 positive. For CD4 positive T cells, we need just to revise the main concept by which CD4 positive or helper T cells act. Helper T cells assist other lymphocytes. For example, it induces activation and the maturation of B cells into plasma cells, which will be responsible for production of antibodies, and also will activate the cytotoxic T cells and the macrophages to attack the cancer cells. And all helper T cells express CD4 antigen on their surface. For the activation of helper T cells, this is very important mechanism. Here, the antigen-presenting cells, for example, mature dendritic cells, will interact with the CD4 positive T cells or the helper T cells by two signals. So in order for the CD4 positive T cells to be active, to be in active state, it needs two signals, signal 1 and signal 2, from interaction with the mature dendritic cell. Of course, first, the mature dendritic cell, how it can be mature, the dendritic cell will take up the antigen, the tumor-associated antigen from the cancer cell, process this antigen, and express this antigen in the form, this red spot. This is the tumor-associated antigen. It will be represented as major histocompatibility complex, and in this case, type 2. So these dendritic cells will process the tumor antigen and will represent or express this antigen in the form of complex with major histocompatibility complex class 2 on the surface of dendritic cells. Here comes the signal 1. So signal 1 comes from interaction of TCR or T cell receptor from helper T cell with the peptide antigen major histocompatibility complex on the surface of dendritic cell. But till now, T helper cell is not active. It needs another signal, which is signal 2. Signal 2 comes from interaction of CD28 from the T helper cell with B7.1 molecules or co-stimulatory molecules, or in fact, we call it CD80 and CD86. All this we will study in details again. But here, signal 2 come from interaction of CD28 expressed by helper T cells with CD80 and the CD86 co-stimulatory molecules expressed by mature dendritic cells. Now, T helper cell become active and started to release some cytokines that regulate and activate all the immune system. That we call helper T cell. On the other side, we have cytotoxic CD8 positive T cell. This is our main player to attack cancer cells. Also, not only cancer, it also destroys virus-infected cells, but here we talk about mainly cancer cells. So all cytotoxic CD8 positive T cells express CD8 antigen on their surface. And here also the activation of CD8 positive T cells comes from interaction with dendritic cells, or generally antigen presenting cell. So antigen presenting cell, after uptake of the tumor antigen and processing of the antigen in the form of a complex with major histocompatibility complex, but here type 1. So for CD8 positive T cells, it must interact with major histocompatibility complex class 1 expressed on the surface of mature dendritic cells. So signal 1 here comes from the interaction between TCR, T cell receptor of the T cell, with the antigen major histocompatibility complex class 2 expressed by the mature dendritic cell. And again, we need signal 2. Signal 2 comes from binding of CD28 by the T cell with B7 molecules or co-stimulatory molecules like CD80 and the CD86 by dendritic cells. When we have the two signals, now CD8 positive T cells are active and they can recognize the antigen of cancer cell and they can attack the cancer cells, leading to their elimination. Here, just this diagram to illustrate that if we have only one signal, the cells are inactive. So if we only have one signal, like the interaction of TCR, T cell receptor from T cell, with the antigen major histocompatibility complex from dendritic cell, and we don't have the signal 2 of the co-stimulatory molecules, so the T cell will not be active, will not be able to attack the cancer cells. So we need two signals in order for the CD8 positive T cells to be active and started to release interferon gamma also to activate other cells, and by themselves they can attack the cancer cells. That's why we call cytotoxic T lymphocytes. So here, if we talk about the key steps required for effective anti-tumor T cell immune response, starts, of course, from death of some cancer cells and release of tumor-associated antigens or new antigens. Those antigens will be handled by antigen-presenting cells, like, for example, dendritic cells. So the dendritic cell will take up the antigen, this red spot, for example, and will process this antigen and make cross-presentation for the T cells. So dendritic cells will go to the lymph nodes and will be in contact with T cells, like we illustrated now. And this interaction with T cells, or what we call priming or activation of T cells. And in this case, T cells start to migrate to the site of action. Here, the tumor will infiltrate the tumor, will go through the endothelial cells of the tumor vasculature and reach the tumor. So here, what we call trafficking of T cells to the tumor, or infiltration of T cells into the tumor. Here, some contact with endothelial cells, because all of these steps we will study in details in our talks about immunoengineering. So after priming of T cells with dendritic cells, T cells will start to infiltrate the tumor and reach the tumor microenvironment and start to interact with the cancer cell. Here, T cells can recognize the cancer cells. Why can recognize the cancer cells? Because it's already activated and primed by cross-presentation of the tumor antigen by means of antigen-presenting cells. So now T cells can recognize the antigen from cancer cells, can interact with the cancer cells, leading to their killing or elimination. How cytotoxic T lymphocytes will kill the cancer cells? By different mechanisms. Also, we will study all these steps in much details, but now just to remember that cytotoxic T lymphocytes or CD8 positive T cells can kill cancer cells by different mechanisms, but here we have two main mechanisms. The first mechanism is the release of some cytolytic proteins like perforin and the granzyme B. Perforin and the granzyme B. Those proteins will be released from T cells when come into interaction with the cancer cell and will be internalized into the cancer cells, leading to their killing or elimination, because these are cytotoxic or cytolytic proteins. We will study more in details, and we will know that perforin form like a channel or a tube that granzyme B can pass through this tube of perforin in the membrane of the cancer cell, and what will happen inside the cancer cell, what is the main mechanism and downstream signals that the granzyme can kill the cancer cells. And also there are another mechanism that T cells can kill the cancer cells by interaction of death ligand, what we call FAS ligand or trail, with complementary death receptors expressed by the cancer cells, leading to their apoptosis. So these are the two main mechanisms that can enable the cytotoxic T lymphocytes to kill the cancer cells. But all these details we will study in our successive talks. So we have different strategies of cancer immunotherapy. First, we can use cytokines like, for example, interleukin-2, interferon, to activate the immune system to start to attack the cancer cell. Or we can use cancer vaccines that we can deliver some antigen, tumor antigens, to activate the antigen-presenting cells to start the immune response and start priming the T cells, the effector organ. Or we can use checkpoint inhibitors, and here is the main cornerstone of our talk. Checkpoint inhibitors, some antibodies that block inhibitory receptors like PD-1 or CTLA-4 expressed by T cells, and reverses the T cells from inactive or suppressed state to active or stimulated state. Or adoptive T cell immunotherapy or CAR T cell therapy, we can take the T cells of the patient out of his body and activate the T cells, or engineer the T cell with some antigens and re-infuse into the circulation of the patient to attack the cancer cells. These are different modalities of cancer immunotherapies that we will discuss in detail. About immune checkpoint inhibitors, what is the idea or the concept by which checkpoint inhibitors work? Here we have the T cells. T cells have some proteins that can activate the T cells, and also have some proteins that can inhibit the T cells upon their binding or activation. So, for example, some activating signal like CD28. We just talk about CD28 is required and essential for interaction with some co-stimulatory molecules on dendritic cell like CD80 and CD86. So CD28, we need to activate this receptor or this antigen if we need to stimulate the T cells. Also OX40 and other receptors or molecules that make stimulation. If we used an agonist for these receptors, we will stimulate or increase the activity of T cells. And this is what we want, right? But there are some also inhibitory signals, like cytotoxic T lymphocyte antigen 4 or CTLA4, or programmed death 1 ligand. And the TIM3 and the LAG3, all these molecules are inhibitory. So if these molecules upregulated or activated, this will lead to inhibition or suppression of the activity of T cells. So cancer cells, of course, tend to upregulate the inhibitory signals. Cancer cells tend to upregulate the inhibitory signals by T cells so that it can lead to inhibition of T cells. This is what cancer cells do. So here is the tumor. Tumor cells bind to T cells and lead to their deactivation. How? By activation of these inhibitory signals or inhibition of these activating signals. What immunotherapy can do? What immunotherapy drugs like immune checkpoint inhibitors can do? They can block the tumor cells from deactivating the T cells. So immune checkpoint inhibitors can prevent or inhibit the cancer cells from deactivating the T cells. How this can be achieved? For example, here we can see that T cell interact with dendritic cells, like we said, and we need two signals for the T cell to be fully activated, right? The first signal comes from interaction of TCR, T cell receptor from T cell, with antigen major histocompatibility complex. here expressed by dendritic cell. If we talk about CD8 positive, so we talk about major histocompatibility complex class 1. And the second signal comes from interaction of CD28 or CD8 expressed by T cells with complementary co-stimulatory molecules on dendritic cell like CD80 and CD86. Here, a mechanism of resistance that T cell can upregulate inhibitory signal from CTLA4. This protein, inhibitory protein, CTLA4, can compete with CD28 to bind with the co-stimulatory CD80 and the CD86, leading to inhibition of T cell. Because in this case, we will lose signal 2. We don't have signal 2. So here, CD28 should interact with CD80 and CD86, but T cell can upregulate, and this is due to the effect of cancer cells, of course. When T cells upregulate cytotoxic T lymphocyte antigen 4, this antigen will compete with CD28 and will inhibit it from binding with the CD80 and CD86 B7 co-stimulatory molecules. In this case, T cell will be inactive. That's why ipilimumab is immune checkpoint inhibitor act by blocking CTLA4. So if we succeeded to block CTLA4 by this antibody, we will reactivate the T cells because we will give the opportunity again to CD28 from T cell to interact with the co-stimulatory molecule from dendritic cell. And here again, we have signal 2 in addition to signal 1, so T cell become reactivated. Another type of immune checkpoint inhibitor, here cancer cell when interact with the T cell, cancer cell need or tend to upregulate PD-L1 by the surface of cancer cell. And this will interact with PD-1 expressed by the T cells. This interaction will lead to exhaustion or suppression or inhibition of T cells. The PD-1, PD-L1 pathway is inhibitory to the activity of T cell. That's why we can use some antibodies to block either PD-1 or PD-L1. Like, for example, nivolumab or pembrolizumab or atezolizumab, all these antibodies act by inhibition of the PD-1, PD-L1 inhibitory pathway. Either we blocking the PD-L1 on the cancer cell or blocking the PD-1 on the T cell. The net result, the T cell become reactivated and can attack the cancer cells. So if we can imagine here, this is just schematic diagram to illustrate the whole concept. When cancer cells treated by any type of therapy, like for example, chemotherapy, because some chemotherapy need to or initiate immune response. When the antigens released from the dead tumor cells, these antigens will be internalized into the antigen presenting cells. The antigen presenting cell will make processing for this antigen and will express this antigen as complex with major histocompatibility complex here, and will interact with the T cells to activate the T cells for priming of T cells. Through two signals, this is signal 1, TCR, T cell receptor, with the major histocompatibility complex with the antigen, and also CD28 from T cell with B7 molecules, CD80 and CD6. But when T cell upregulate CTLA4, this will lead to inhibition of T cells because this CTLA4 will compete with the CD28 and will bind with the B7 protein, and we lose signal 2, so T cell will become inactive. Here we use the ipilimumab to block the CTLA4 and give the opportunity again for CD28 to interact with the B7 molecule CD80, CD86 on antigen presenting cell, and T cell become active again, will infiltrate the tumor, will interact with the tumor cell, leading to their killing. However, the cancer cell upregulate PD-L1 and PD-L2, and those inhibitory molecules will interact with the PD-1 protein on the surface of T cells, leading to their inhibition. So if we use the anti-PD-1 antibody, for example, this will block the PD-1 antigen, so inhibit the PD-1, PD-L1 inhibitory pathway, and T cell become activated again and kill the cancer cells. So we have some challenges of cancer immunotherapy. Although the great success achieved by cancer immunotherapy in certain types of cancer, but still we have about 60% of cancer patients fail to respond to immune checkpoint inhibitor. Those patients develop resistance against cancer immunotherapy. What is the reasons of this resistance? This we will study in detail. And this is in addition to the serious non-specific systemic inflammation and autoimmune side effects that can occur when we use the immune checkpoint inhibitor. And here comes the importance of engineering tools like nanomedicine for targeting these immunomodulatory agents to the tumor microenvironment to avoid the side effect on the peripheral tissues. So if we can summarize the mechanisms of resistance to immune checkpoint inhibitors, we have three main factors. Number one, low immunogenicity of cancer cells. So cancer cells tend to be non-immunogenic, like we mentioned before. They can down-regulate the expression of tumor antigen on their surface. So they can't be recognized by antigen-presenting cells. Or cancer cells can render the dendritic cells with immature or have impaired antigen-presenting capacity. So although the cancer cells immunogenic, but dendritic cells sometimes are tolerant or immature or have weak or poor or impaired antigen-presenting capacity. They can't cross-present the antigen and cannot prime the T cells successfully. Or also cancer cells tend to generate immunosuppressive tumor microenvironment. So cancer cells can release some cytokines, some factors, some chemokines that modulate the activity of other cells in the tumor microenvironment, like the cancer-associated fibroblasts, the tumor-associated macrophages, myeloid-derived suppressor cells, T regulatory cells. And all these have immunosuppressive role. What is the meaning of immunosuppressive role? That all these cells will suppress the activity of cytotoxic CD8 positive T cells. So those are the main mechanisms of resistance of immune checkpoint inhibitor. It can be illustrated again by this schematic diagram. So here, for example, the tumor cell or antigen presenting cell in their interaction with the T cell. If cancer cells succeeded to downregulate the expression of major histocompatibility complex or their tumor-associated antigen, we will lose signal number one that needed to be interact with T cell receptor. In this case, T cell will not be active. Or alternatively, tumor cell or dendritic cell or antigen-presenting cell can downregulate the expression of co-stimulatory B7.1 molecules, CD80 and CD86. Although signal 1 is present, however, we lose signal 2. CD28 from the T cell will not interact with the co-stimulatory molecule on the antigen-presenting cell. We lose signal 2, again, T cell become inactive. Or the interaction or overexpression of PDL1 by cancer cell or antigen-presenting cell will be interacting with the PD1 on the T cell, leading to their exhaustion or T cell inhibition. Also, cancer cell can secrete some factors like chemokines or TGF-beta, indole diaminoxygenase, some enzymes, some factor, vascular endothelial growth factor, some cytokines, interleukin 10 and interleukin 35. All this can modulate the activity of tumor microenvironment and generate immunosuppressive tumor microenvironment. This will affect the activity of T cells. So from here, we will talk about the different cells present in the tumor microenvironment. So we have immunosuppressive tumor microenvironment cells that can suppress the activity of T cells. In fact, what is happening? Cancer cells tend to switch the tumor microenvironment from immunoresponsive or immune hot to immune cold or immunosuppressive tumor microenvironment. For example, the fibroblast, by the action of cancer cells, will be differentiated into cancer-associated fibroblasts, or what we call CAF, and these have inhibitory effect on the activity of T cells. The same for macrophages. Cancer cells tend to repolarize the macrophages from M1 phenotype to M2 phenotype, and M2 phenotype has pro-tumorigenic effect. They can help the cancer cells to grow and and metastasize. And also cancer cells tend to make the dendritic cells from active state to inactive immature state, or also the neutrophils from N1 to N2. So in conclusion, or overall, that cancer cells tend to remodulate the tumor microenvironment into immunosuppressive. And here are the main immunosuppressive cells in tumor microenvironment: regulatory T cells, myeloid-derived suppressor cells, M2 tumor-associated macrophages or TAMs, cancer-associated fibroblasts. So all these cells we need to reprogram again into immunoresponsive phenotype. So how to overcome those challenges, how to overcome the development of resistance against immunotherapy or immune checkpoint inhibitor? We need certain immune mechanisms and novel tactics for increasing the immunogenicity of cancer cells and reprogramming immunosuppressive tumor microenvironment. Those are the two main factors we need to handle to reverse the immunosuppressive tumor microenvironment to immunoresponsive or immunostimulant state. And we also need to increase the immunogenicity of cancer cells. So what we will study in immunoengineering strategies to help enhance the efficacy of cancer immunotherapy. Some immunoengineering strategies are targeted to enhance the cancer immunogenicity, and some are targeted to enhance the antigen-presenting capacity of antigen-presenting cells like the dendritic cells. Some other strategies we will study, we will target to make metabolic reprogramming or epigenetic reprogramming of tumor microenvironment. Also, we will do some immunoengineering tools to enhance the phagocytosis of cancer cells by macrophages. We will also use engineering tools to reprogram the stromal cells like the cancer-associated fibroblasts. We will study also some cell-based immunoengineering like CAR T cells, for example, and the new trends of immuno-oncology. This will be our main content, how we can use the immunoengineering tools to remodulate the immune system, to activate the immune system to fight the cancer cells. So just to summarize, we will use some engineering tools like nanomedicine, for example, to repolarize the tumor-associated macrophages from M2 phenotype into M1 phenotype. We talked about this briefly, and we said that M1 macrophages have anti-tumoral phenotype. M1 macrophages have the ability to phagocytose the cancer cells and activate the T cells, the T helper cells, and also activate the cytotoxic T lymphocytes. And themselves, the M1 macrophages can kill the cancer cells by different mechanisms, like, for example, generation of reactive oxygen species. However, cancer cells have the ability to repolarize of macrophages from M1 antitumoral phenotype to M2 protumoral phenotype. So cancer cells secrete some factors here, like colony-stimulating factor 1, that interact with complementary receptor on macrophages. This will lead to repolarization of macrophages from M1 to M2. These M2 have protumoral phenotype. That means have immunosuppressive effect on the T cells. They can inhibit the T cells by secreting some factors, for example, interleukin-10. Also, these M2 macrophages can activate the regulatory T cells that have immunosuppressive effect. They can also make breakdown of extracellular matrix that help the cancer cells to metastasize. They can also help the angiogenesis and formation of new blood vessels for the growth of tumors. So here, what will be our target, immunoengineering target? Our target will be the design of certain immunomodulatory strategies that can repolarize again the macrophages from M2 to the antitumoral M1 phenotype. We will study all these immunoengineering strategies. Also, we will study some immunoengineering strategies that can reprogram CAFs or cancer-associated fibroblasts into the quiescent fibroblast phenotype. Cancer cells also tend to reprogram the fibroblasts present in the stroma of the tumor into the immunosuppressive cancer-associated fibroblasts. Like you see here, cancer-associated fibroblasts secrete different factors to modulate the whole tumor microenvironment. They can suppress the activity, of course, of the cytotoxic T cells, like you see here. They can also activate the M2 macrophage phenotype here. So CAFs can also help polarization, like the cancer cells of macrophages from M1 to M2. They can activate the T regulatory cells. They can induce inactivation or immaturation of dendritic cell. This will lead to reduce the activity of T cell again. So CAFs have major immunosuppressive effect. So our target here, using some engineering tools, is to reprogram the CAFs from the activated immunosuppressive phenotype into the quiescent fibroblast phenotype that can antagonize the activity of cancer cells. Also, the dendritic cells, we will use some engineering tools to maintain the dendritic cells in its mature, activated, antigen-presenting state cell. So the maturation of dendritic cell will help to activate both CD4 helper T cell and also CD8 positive T cells. However, cancer cells tend to induce tolerance or resistance of dendritic cells, and in this case will not be able to activate the CD4 or CD8 positive T cells. So our target here is to maintain the dendritic cells in active state, in mature state, so they have high antigen presenting capacity and activate both types of T cells. Also, we will use some engineering tools to inhibit the activity of T regulatory cells or Tregs. Tregs have immunosuppressive action by different mechanisms. Either direct contact with dendritic cell, making inhibition of the dendritic cell. Also, they can release granzyme and perforin, in this case, to kill the CD8 positive and CD4 positive T cells and suppress the immune response. Also, T regulatory cells secrete some factors like interleukin 10 and 35 and TGF-beta. All these factors have immunosuppressive effect on both types of T cells. Also, T regulatory cells generate adenosine, that is a known immunosuppressive factor that can inhibit the activity of T cells. We will study all these mechanisms in details with the engineering of Tregs. Some new strategies we will talk about also is generation or engineering of antibody drug conjugate. This is also under the umbrella of immunoengineering. This type of antibodies is conjugated. This is antibody targeting the cancer cell, antibody that can bind certain antigen overexpressed by cancer cell. And from the other side, it is linked to cytotoxic drug or cytotoxic therapy by certain linker. So this is called antibody drug conjugate, and this will ensure that our cytotoxic chemotherapy reach the cancer cell. But here by means of antibodies, that's why it's also studied under umbrella of immunoengineering, because we use monoclonal antibody. Also, another novel strategy is use of bispecific T cell engager, or what we call BiTE. This type of antibody it can bind to T cells and the cancer cells in the same time. So it has some fragment specific for CD3 on the TCR receptor of T cells, so it can bind specifically with T cells. On the other arm, it can bind with a certain tumor-associated antigen in the or expressed by the cancer cell. So this BiTE or bispecific engager can bring both T cells and the cancer cells into close proximity, creating what we call immunogenic synapse. And here T cells will start to release the perforin and the granzyme B and start to kill the cancer cells. Another strategy, of course, is the CAR T cell therapy, and we use different immunoengineering tools to engineer the surface of T cells to be able to fight and attack the cancer cells. In summary, we can remove the blood from the patient and isolate the T cells using certain kits, then transfect the T cells with certain gene or DNA, including chimeric antigen receptor, CAR. This chimeric antigen receptor can bind specifically to tumor-associated antigen expressed by the cancer cell. So we here need to induce expansion and proliferation of CAR T cells in vitro. Then we collect these T cells and reinfuse the CAR T cells, the engineered T cells, into the patient again. And here we activated the T cells and increased its number and activity to be fully active and be able to attack the cancer cells. We can also use some engineering tools like nanomedicine to engineer the surface of T cells, not only induce the expression of CARs. This we'll also study in details. Also, we can make what we call biomimetic. So, for example, nanoparticle, we can decorate the surface of nanoparticle with some antibodies, and we can also decorate the surface of nanoparticles with cell membrane proteins isolated from immune cells. So, for example, if we need to make biomimetic to CD8 positive T cells, we bring the CD8 positive T cells, and by certain kits, we isolate the membrane proteins in active state and take these membrane proteins and use to coat the surface of nanoparticles. That we call biomimetic. So here we coated the surface of nanoparticle with the actual membrane proteins present in the immune cell. So here the nanoparticle will act as T cells. That's why we call engineer biomimetics. Also, we will study engineered immunotheranostics. What we mean by immunotheranostics? Theranostic, that means they have therapeutic agent, the immunomodulatory drug, and also diagnostic agent, some agent to make imaging, to be able to or to enable imaging of the tumor to monitor its progress. And another type of immunotheranostic advanced generation is immunoreporters. So, for example, here, if we use immune checkpoint inhibitor, anti-PD-1 or anti-PD-L1, this will activate the T cells. And T cells, like we mentioned in the mechanism, will release granzyme B. So here, for example, this nanoparticle have some dye and the dye quencher connected together by certain bond. This bond or peptide or amino acid sequence is sensitive to granzyme B. So let's say we have this immune checkpoint inhibitor activated the T cells to release the granzyme B. In this case, granzyme B will break the bond between the dye and its quencher, releasing the dye in active form, and the dye will induce fluorescence, induce imaging. This can give us report about the activity or sensitivity of the tumor to immune checkpoint inhibitor. However, if this tumor is resistant to immune checkpoint inhibitor and we injected the anti-PD-L1, T cells will not be activated if they are insensitive, if cancer cells can inhibit the T cells. So no granzyme B will be released. So the bond connecting the dye and its quencher will not be cleaved, and the dye will be in inactive state. No fluorescence will be detected. So we can realize that this tumor is resistant to the immunotherapy. This is what we called immunotheranostics. Of course, I will illustrate all these mechanisms again in details. This is just introductory lecture to let you know what are the immunoengineering tools that we will study in the successive lectures.